Good afternoon, ladies and gentlemen. My name is Marek Michalewicz. I'm the organizer of this series of virtual ICM seminars in computer and computational science. And I welcome you all to the 21st seminar in the series. I need to acknowledge great support of different parties which help us in promoting this event and support us. First of all, our University of Warsaw, National Information Processing Institute, and then several publishers of HPC and computer information. It's HPC-Wire, Datanami, Enterprise AI, ITWIS, and Forum Akademickie. This is the 21st seminar. So far we had 20, and the numbers are quite good. We had, including today's speaker, we had 21 outstanding scientists speaking about their research. There were over 1,600 participants at those seminars. There are over 7,000 views on YouTube channel where we present all our seminars, and I encourage you to see some of those seminars that might interest you. And there are altogether over 800 hours of viewing at YouTube. Plus, this is a sort of additional activity, additional to what we do annually, which is Supercomputing Frontiers Europe Conference. And the traffic at that website is also quite impressive. We had 5,000 over nearly 6,000 returning visitors. And I would encourage you, if you're interested in today's topic, I would encourage you to go and look at editions from 2019 and 20, where we had special invited speakers from Henry Markram group on connectome research. We had Leon Chua talk about memory stores, and that's a very interesting sort of avenue of research into physics-based models of neurons. And then in 2020, we had Jim Jimczewski talking about his research, experimental research on completely synthetic bundles of neurons, which is maybe far away from what we will be hearing here, but perhaps one day those avenues of research will merge somehow. Who knows? And then the previous 20 speakers are all outstanding researchers in their own fields. And here's the full list. I won't be reading all those names, but you, of course, welcome to browse to this link here given at the bottom. And there are some really fascinating talks. All of them are great. Not all might interest everybody, but of course you are free to choose. And I welcome you all to go and see. And today is my great pleasure to welcome Sven Dorkenwald, who is currently a PhD student in Professor Seung's lab at Princeton University. In his PhD, he's developing systems, infrastructure, and machine learning methods to facilitate the analysis of large-scale connectomics datasets. Together with collaborators at the Allen Institute of Brain Science, he developed proofreading and annotation infrastructure that is used to host multiple large-scale connectomics datasets and runs FlyWire. FlyWire AI is an online community for proofreading neural circuits in the whole fly brain based on the FAFB EM datasets. And with this, I welcome Sven, and the screen is yours, Sven. Thank you very much.
Thank you very much for the kind introduction. I'm excited to tell you about our effort to reconstruct the connectome of the whole fly brain. So let's jump right in. What I'm showing, what you're looking at here is a cut through an entire fly brain acquired by Sheng et al. In David Bock's lab at Geneva Research Farm. This electron microscopy image is at a very high resolution, so that when we zoom in by about 100x into this brain, we can appreciate the fine ultrastructure of the neurons in the synapses within it. So here I'm going just over a few points that you can see. First, membranes. Membranes surround neurons, and everything that's surrounded by a membrane is usually a neuron. Then vesicles. So these blobs hold neurotransmitters and are usually found close to a synapse. And then finally, T-bars, which are found close to most synapses in a fly brain and usually indicate a synapse. So when by coloring this in here, in red, I'm showing you the presynaptic neuron, which is talking to the colored ones on the postsynaptic side. And because of 3D data set, we can now follow these neurons throughout the entire brain. Here I'm showing you what this looks like on the whole brain scale. And even though we are only seeing a small section here, you can appreciate how these stretch through the entire brain. And then in a second, we will see for just this red neuron, where it is talking to other neurons. So in blue and in green, here I'm showing you all the synapses where it is receiving input from other neurons and then also talking to other neurons in return. So with this method of starting from an electron microscopy dataset, then reconstructing individual neurons, finding the synapses between them, we are able to construct wiring diagrams of any number of neurons in this brain. So in such a wiring diagram, every row is a presynaptic neuron talking to any number of postsynaptic neurons on the y-axis. And in this diagram, the color indicates the number of synapses. This particular one was created by Krista Baker for about 500 auditory neurons in the fly brain to study sensation in the brain. More broadly, though, the goal that we have with these wiring diagrams is to gain a fundamental understanding of how neural circuits are built and how they give rise to function and behavior. And of course, that comes with computational challenges. Here in this talk, I'm going mainly about 3 of these challenges, which are how we assemble these 3D data sets in the first place, such that they are amenable for automated reconstruction, then how we create these neuron segmentations, and then finally, you will see those neuron segmentations need further correction, how we can then actually go and proofread these neurons such that they can be used for analyses. The first connectome was acquired for this very little animal, C. elegans, a nematode about a millimeter long. It has 300 neurons, 102 to be precise. And this effort was led in the 80s, and it was a truly heroic effort, took multiple years. But the methods that were developed back then, right, the systems, they're still roughly used today. And what they did is they stained the sample with heavy metals such that they are visible under an electron microscope, fixated it in a resin, and then cut it into thin slices, so thin that when you assemble them, you can have basically a 3D data set. You image these individual sections, and then you build your 3D data set. So while, of course, technology has progressed and automation has set in, it's still the way how we build these data sets today. The connectome of the C. elegans has about 5,000 synapses, and that is in contrast to, for instance, a whole fly brain, which has about 150 million synapses. And instead of 302 neurons, the fly brain has about 100,000 neurons. And to get to a brain like the fly brain, we had to scale up these methods of acquiring EM datasets. And to give you an idea for why scale is so important, here I'm overlaying different sized volumes onto those same neurons that we started with initially. And I hope you can, in this video, and it plays in a second, I hope you can appreciate that in these tiny volumes, we may be able to encapsulate an arbor of a neuron, but to truly get a sense of the whole connectivity of a neuron, we have to acquire a larger and larger volume. And even though we can find synapses in small data sets, we may also often not know the identity of a neuron that we see in a data set, because we may need the entire neuron to appreciate that. So that is part of the reason for why the field of connectomics has worked hard to scale up the size of these data sets that can be acquired. And what I'm showing you here is every dot is an individual data set in its size on a log scale. And over the last decade, data sets have been increased in size dramatically. And on the upper end here, these two, they're the first data sets that are about in a cubic millimeter scale, and each of them takes up about a petabyte of storage just for the raw EM data. So while EM acquisition is still a huge challenge and something that needs to be scaled further, with this increase in scale, of course, another problem set in, which is how do we analyze this data? Originally, most of these data sets were analyzed manually. And I will show you in a second what I mean by manually, but increasingly, we see that these data sets are analyzed with semi-automated methods, where we first introduce some sort of automated segmentation that we can manually correct. So classically, these data sets were reconstructed using skeletonization, and what I'm showing you here is a video from WebNozzles, which is a platform that can be used to host EM data sets and then skeletonize neurons within them. And here, one tracer is essentially flying along through the neuron, leaving skeleton nodes behind as they progress through the slices. And here, coming to a branch point, may leave breadcrumb behind, following one branch, coming back to the other. But obviously, there is an upper limit of how fast you can go with such a method. There's an upper limit on how fast a human can go, and the only way then to scale is, of course, to add more traces. But I'm showing you here is one of the largest data sets that have been reconstructed this way. But because we are limited how much we can reconstruct, only a certain number of neurons within such a data set may be reconstructed. And also, just as an anecdote, usually this has been done with many part-time students, and that's also how I started actually in this field 8.5 years ago, reconstructing and skeletonizing neurons in a zebrafinch data set at the Max Planck Institute in Heidelberg. So for the latest data sets, though, this method is just not able to reconstruct neurons on a whole scale. What I'm showing you here are the latest, largest data sets. On the left is a sample from a human data set, and on the right, from a mouse visual cortex. This was acquired by the Microns consortium, of which the Sang Lab was part of. But throughout this scaling up process, also of the methods, what happened is that we essentially also surpassed the entire size of a fly brain. So our methods for reconstructing neurons are now able to handle a whole fly brain, and that opens up a whole lot of opportunities. To talk briefly about the Drosophila, which may not sound initially as interesting as a mouse or a human sample, the Drosophila itself shows a vast variety of behaviors that have been extensively studied. I'm just showing you of two here. So here on the second one, you can see a smaller male fly following a bigger female fly. And quoting here, you can see how they're flapping their wings. That's basically a way of singing and communicating. So there's extensive behavior that has been studied by a great community. And because we are now able to handle the whole fly brain and reconstruct neurons within it, we're in a position where we can reconstruct circuits that are mostly complete and can relate them to these studied behaviors. Also, because invertebrate brains are stereotypic, meaning that if you have two brains, you may find the same neurons, at least the same neuron types. In both of them, you can try to find, like Christa Baker did here, to take a reconstruction from one data set, like in the EM data set, and find its equivalent in another imaging modality, like a light microscopy data set. And that allows us to tap into all kinds of other resources that the field has built up and annotate a connectome that we might extract. And because of all these benefits, connectomics has been used in the field of Drosophila for quite some time. I'm showing you here two large resources. The upper left is a community, an effort to reconstruct, manually reconstruct all neurons in a Drosophila larvae. So that has some thousands of neurons, and the effort is slowly coming to an end, I believe. And on the upper right, a very popular one that's also a semi-automated approach where a part of a brain was imaged and reconstructed, and that was then proofread by 50 person years of effort and released in 2020, and has set standards for this kind of data. This paper that is associated with that has seen over 170 citations and enabled all kinds of other research into the circuits of the Drosophila. Though at the same time, another resource, the dataset that I have been showing you before, the FAFB full adult fly brain EM dataset, has been published, but so far has only been reconstructed manually. So we have in the SciLab seen an opportunity to contribute not just a part of a brain, but a whole fly brain reconstruction that can then be studied by the field. So we started on a journey that it has now become FlyWire to reconstruct all the neurons in a fly brain. And to enable that, we had to overcome three broad challenges, and that I will now be going over one by one. The first one being the assembly of the initial 3D stack. You will see in a second why this is so challenging, but essentially if you are making a mistake in the assembly of this image stack, you will introduce errors that you won't be able to correct in the later processes of the pipeline. So this is an extremely crucial step because it will set the stage for all steps to come. Then, of course, automated segmentation, reconstructing all the neurons in this brain, and finally proofreading. And in our case, proofreading by community, and that's also the point on which I will spend most of the time on. The FlyWire image dataset, the FAFB, was acquired with an electron microscopy method called transmission electron microscopy. And this method was thought to be less amenable to automated segmentation. And the reason for that is that in this imaging method, slices of a brain are collected on a tape. And because that is more difficult to see on the second image here, I'm showing you a slightly better video from a different data set by the L Institute TEM team. Here in the back, there's the image data, there's a data set mounted onto an arm that is going down and cutting off a slice on a diamond knife. Then this ultra thin section of about 40 nanometers thick is then collected on a tape, and you can see slots. You have to hit those slots perfectly, and then spooled up and then passed into one or multiple microscopes. But in this cutting process, because there are all these forces on the stack, these images tend to be deformed, and that makes the assembly process later very difficult. To give you an idea for how a well and not so well aligned stack looks like, here's a series of images of what we would call a rough aligned dataset. It's extremely difficult to follow any neurons through these slices. And when we finalize this dataset, you are now able to follow individual neurons as we pass through the stack. And we can judge a alignment, a assembly, by how correlated neighboring sections are, because we expect a smooth flow through the dataset. what makes this even more challenging for images from this microscopy method is that there are special kinds of artifacts that are introduced and that introduce non-smooth deformations in the images. And I'm showing you two of those here. What can happen in the imaging process is that a section folds onto itself. So essentially, we are losing some of the image below itself, and that is then shown by a black stripe in the image. Now, while this data is lost, that is below the slab, the main problem is actually that the whole image has been pushed together dramatically, and in the alignment process, we have to pull it apart again. And the inverse process is cracking, where an image is cracked apart, and we have to now push it together to correct for this. Now, these non-smooth deformations kind of introduce singularities in this alignment process, and that has been traditionally very difficult to handle by state-of-the-art alignment method. On the lower left, I'm showing you a cut through the brain, but this time you can also see that there is some artifact running almost through the entire brain. And in the method that was used to align the original EM, and when we analyze that by judging the correlation at every location with the neighboring slice, we can see, so basically one is obviously high correlation shown in white, and then in black we have very low correlation, and this one artifact here is introducing not just very low correlations at the artifact itself, but in the surrounding area, because it's very difficult to handle the alignment surrounding this area. If there are artifacts on every 10th or so slice, that means that many images are affected by that. And eventually a neuron reconstruction, a neuron might run into one of these and then therefore be difficult or impossible to reconstruct correctly at that location. So it's paramount to have a very good alignment to start with. And to handle such cases, a group in the Sung Lab led by Sergej Popovich and Tommy Macrina developed essentially a completely new method to aligning these data sets. And this method uses CNNs to essentially predict how one image should be transformed to match the next image in the series. So we can always use two consecutive images and ask, how do we have to change the one to make it look as close as possible to the next one? And the method that they've developed based on a CNN is producing what they call displacement fields, and these are essentially vector fields. So we can take then one image, calling it source, and then apply this vector field to make it so that it's very close to the next image in the stack. And if we do this sequentially through the entire stack, we hopefully have a well-aligned stack where two neighboring images are very similar. To give you some more intuition for that, what I'm showing you here is an EM image without any artifact, and overlaid are in color the vectors that were used to transform this image to this location. And what one can appreciate here is that these colors are very similar, meaning that neighboring pixels were transformed in a similar way. However, at a location where there is an image artifact, these vectors are now able to change very rapidly in a very small span, such that now on the left, all these pixels have been pushed to the left, and on the right, they have been pushed to the right, and this way we can handle these rapid image artifacts. Because this is essentially a pairwise method, we can now scale this to a large data set by parallelizing the alignment of neighboring sections. So we can take pairwise pairwise slices, align them to each other, and then complete an entire data set. To give you a sense for the scale, I estimated that for the FlyWire data set that's using FAFB, this took about 15,000 GPU hours, but for the latest petascale data sets, this may take some hundreds of thousands of GPU hours. So to show you that the result for this specific section here, where we had a big area of misalignment before, we are now able to produce only a very small area that is just basically the area of the actual artifact, and recover most of the image surrounding this artifact. And then with this image newly aligned, we are now left with the task of actually segmenting this image. Segmentation is then the problem of coloring in the neurons. So for every neuron, we want to assign it an individual ID indicated by a color. And this problem is very similar to other image segmentation problems in computer science. I think the biggest challenge here is that it is actually in 3D, and those neurons are very intertwined. On the right, every color is a different neuron ID, and you can see these are two adjacent slices, and the same color indicates the same neuron ID. We don't actually go directly to individual IDs. The intermediate step is what we call an affinity map. So here we predict for every pixel how likely it is that the adjacent voxels belong to the same object, the same neuron. And we do this in all 3 dimensions. And this is color-coded here in X, Y, Z. So areas that are shown yellow, they have a high affinity, meaning they belong together in X and Y, but have a lower one in Z. And this can then be seen as some sort of landscape that we can then basically fill up. The standard method for that is a watershed. We basically fill up to what we call supervoxels, very small fragments, but we can treat them as individual pieces that then make up the bigger segmentation. In this step, we are very conservative, such that we can treat them later as essentially atomic pieces, and then combine them together to a whole segment in a process called agglomeration. And this process is, and we can parallelize this process essentially through an entire data set by cutting the data set apart and processing individual chunks individually on each one on the GPU, and then assembling back together to a big data set. The Sang Lab has developed multiple workflows for that. The one that we used the most probably is ChunkFlow, which is a public workflow pipeline that can be used from the Sang Lab repository, and it's able to run these kinds of tasks on all kinds of cloud provider platforms and locally, wherever one wants to run these jobs. So combining that all together, we were able to build a new realigned data set of the FAFB imagery. Here on the left, I'm showing you this image, and as we scroll through, you can appreciate that it's a very smooth scrolling. We can follow individual neurons, and we can color in individual neurons here on the right, and colors, as we flip through, they don't keep changing on us. We have applied this to the entire data set, meaning that every voxel in this data set is now assigned a neuron ID. And these segmentations are best judged, though, in 3D. So here I'm showing you example neurons before any proofreading was done. So these are fully automated neurons, how one would find them when going into FlyWire before proofreading. And we compared some of them to light level reconstructions of the same neurons. And because the FlyBrain is quite stereotypic, we expect them to look almost the same. So here, for 2 very popular neuron types, the giant fiber and the APL, we found other images of these neurons to compare and see that our method is not missing any arbors systematically. And I hope you can appreciate that these look quite similar, though there are definitely things that are a little bit different. But overall, these automated segmentations are good enough that a neuroscientist can identify them as their cell type before any proofreading. And that is important, as you will see later. Now, with proofreading, those neurons become more complete, and proofreading is necessary for analysis. Unfortunately, these segments are in the automated segmentation not good enough to be used for analysis, but proofreading is actually quite fast. You will see later it takes about 10 to 20 minutes for an experienced proofreader to proofread a single neuron. However, on a whole brain, this is still a lot of work. And that kind of creates some kind of dichotomy where we don't want to wait until we have proofread the entire brain and then only read it then to researchers, because even just proofreading a few neurons enables a whole lot of analyses. So we combine these insights essentially to creating a community for researchers that together proofread neurons in the FlyBrain. And by that, every researcher can proofread neurons of their interest, use them for analyses, because even subconnectomes can be extremely valuable for analysis, but thereby they also contribute to the proofreading of the entire brain. And that is essentially what FlyWire is. FlyWire is a community of scientists that together proofread the brain by proofreading their neurons of interest. FlyWire is a platform where everybody sees the latest state of the segmentation. So here I'm showing you just a number of them, and everybody sees each other's result in real time. And of course, on some level, that's also a social experiment, but the Fly community has been great and quite used to these kinds of communities. And I think from a social side, that is going very well. However, we had to come up with new computational tools to facilitate this kind of proofreading of neurons on that scale by scientists all over the world. And prior to FlyWire, usually proofreading of neurons has been done in a more asynchronous way. There, for instance, for the hemibrain dataset that I have shown you before, there has been a centralized effort with proofreaders to proofread neurons that were organized by a proofreading scheduler. Usually data is co-located to these proofreaders to make visualization updates easier. And then only very infrequently, all these changes are packed together and then materialized into an updated segmentation that then can be used for analysis. But as I alluded to before, that doesn't really work for us in FlyWire because we can't control what every scientist wants to proofread. And in an asynchronous workflow, analysis is delayed until after proofreading is complete. But here we want to allow people to proofread the data whenever they are done proofreading. So essentially, we had to come up with a new data structure that facilitates this kind of proofreading analysis that are going on in parallel, and that was part of my PhD work to this date. So the work that we have been doing for proofreading builds on these supervoxels that I have shown you earlier. So supervoxels are very small groups of voxels, and we built essentially a graph of these supervoxels. Wherever they touch, we insert an edge. And now this transforms the segmentation problem to a graph problem. In this graph problem, each neuron is a connected component of supervoxels. And the initial segmentation, instead of producing a voxel segmentation, is essentially producing an initial set of connected components from the supervoxel graph. In this graph world, proofreading is not a changing of individual voxels, but rather a change of the graph. So if somebody would like to merge two components together, what they would do is they insert an edge in this graph, and thereby creates a new connected component that is a combination of the initial two components. And if they want to split something apart, they would remove edges from this component, creating two new components. So this graph is very large. In FlyWire, our graph has about 2.3 billion supervoxels and about 10 times as many more edges. And in these petascale data sets, we are reaching trillions of supervoxels and edges. Each individual neuron itself can also be quite large, having tens of thousands of supervoxels to millions of supervoxels that make them up. So essentially, the challenge is now, how can we make graph changes rapidly to facilitate proofreading in real time? And the main insight that we had is that even though proofreading is always affecting an entire neuron, edits are usually quite local. So what I'm showing you here is a whole neuron after proofreading, and in black. And in color, there are segments that have been removed. Here in this image, I'm zooming in onto one of such locations where an arbor has been removed from the main neuron. And that proofreader, when they made that decision, they only locally made an edit, made a split of the segment, but the effect propagated through the entire neuron. Still, nothing changed actually in the rest of the neuron. So if you can just cache the connected components of these other parts of a neuron, we are able to scale to these large graphs. And to show you how we did that, here are some visualizations. So we spatially chunk each neuron into a grid, essentially an octree in 3D. And as we combine these individual chunks of the grid, we build a hierarchy of chunks that get larger and larger. And when we reach the top of this tree, the top of this agglomeration of these grids, we reach an entire neuron. And to visualize this a little bit differently, here on the left, I'm showing you what a naive version would look like. Here we have individual supervoxels shown at the bottom. And because we want to store the connected components of these supervoxels as they're connected with these edges, we would just, in the naive version, just all have them point to one parent node. In the method that we now implemented, we build essentially an octree on top of this graph. So we fuse together the concept of an octree with a graph. And that has the benefit that each of these nodes here, as we go through the layers, resembles a connected component in the underlying graph. So at first on layer 2, this one node here combines all the supervoxels in a chunk to one connected component. And when merged together with adjacent chunks, each node higher in the tree resembles a bigger piece of the underlying neuron. Now, that can be quite abstract, and to show you what this looks like in practice, I'm essentially now doing two tree traversals. First, by clicking on a supervoxel, so here on the right we see the supervoxel segmentation, we're going up this tree hierarchy. So from a supervoxel, we will now retrieve the overall neuron, the root of this tree, and then from there, going back down, retrieving all the supervoxels that are within the chunk, and then coloring them in the same color. So here I'm clicking, and that's all it took. We went up the tree, we retrieved the neuron ID, which is shown here on the right, and we go back down this tree and retrieve all the supervoxels we have to color in the same color. And I hope you can appreciate that this is actually quite fast, even though in FlyWire our tree has about 10 levels. And we're able to do that quickly because our system, the chunk graph, is implemented with Bigtable that has extremely low latency random row reads. To give some numbers to this experience, These are measurements from the initial phase of FlyWire, and this process of going from leaves to roots in our tree on the server side is taking us less than 100 milliseconds, and the same goes for going from the top to the bottom of the tree. And the benefit here being that we can we can restrict what we retrieve to the area that the user is looking at, and we don't have to retrieve the entire list of supervoxels. So far, that might look like some fluff added to some graph structure, but the benefits become more evident when we actually want to do edits in the structure. So for an edit, to remind us, we want to remove edges from this graph and then compute new kinetic components. And to do that on a whole scale, we would have to load all the supervoxels, all the edges between them, remove the edges, and then recompute kinetic components. In the chunk graph system, we don't have to load the entire graph. We only have to load the local section that is affected. And here on the left, I'm depicting this with this red edge where we removed an edge. We loaded the local graph in this chunk, computed kinetic components in the chunk itself, had new kinetic components resembling two new parents. And now from there, instead of having to compute kinetic components in the rest of the graph, we can now fuse these new components on level 2 with neighboring chunks, with kinetic components from neighboring chunks as we go up the hierarchy. So as we go up the hierarchy, we now in the end create a new root for every component that comes out of this split. And additionally, because we can assign timestamps to these edges, we can version our tree in time continuously. So we can always go back in time, use a timestamp that was earlier than this initial timestamp T0, and we can go up the original tree, we can retrieve the original neuron, and we can use a later timestamp or the latest timestamp to go up the latest tree. And to put some numbers to this, we compared first reading edges. So I mentioned before, we only had to read a subset of edges from this graph to make that happen. And unsurprisingly, we are much faster than reading the entire graph. But this is also showing how intractable the original approach was, because reading all the edges, even with a system like Bigtable, takes us about 10 seconds, which is absolutely too slow for a really interactive system. And so combined, merges and splits take about a second for a merge and about 2.5 seconds for split, which is within the realm of what people are happy to wait for when they command and edit. So then to give you an example of what this looks like in FlyWire, here I'm now doing a merge operation. I have this neuron. I noticed it is too short. That comes with experience, and I'm looking for an extension. Now I can zoom in, and indeed, one can appreciate that this neuron continues through this originally false cut in this turquoise segment. I can now command a merge by clicking on each side, and that's all it takes. The system on the back end now combined these two segments together, and I want to go back to this, and created this new yellow segment. Every user that will now click on the segment from now on will see these the updated segmentation. What you can also see here is that there is a little hole left at the location where we made the edit, and that is because we have to now go and update the visualization at that location. But because of how we're doing meshes, we can show the mesh at least for the rest of the neuron. And what we're doing here is we are creating meshes, and that's that's a rendering of that for every single node in the hierarchy. So here I'm showing you meshes for what we call level 2. That's the first level where we agglomerate supervoxels together. And every piece here is a different color because that is an individual mesh. And the whole neuron is then stitched together. So as we go up the hierarchy, we stitch together these small meshes to make them to a big mesh. But when we have a proofreading edit, we only have to actually recompute that mesh in that very local area and then can just stitch it together with the neighboring meshes. And that makes meshing much, much more efficient. And we can usually update visualizations within 5 to 10 seconds. Okay, and with that, that basically wraps up the computational side of FlyWire. The community has been going strong. I'm showing you here some numbers over the last 2 years. We opened up FlyWire to more and more labs, starting with an alpha and a beta phase. And here in the upper plot, I'm showing you the number of unique users that have been making edits in FlyWire. There are much, much more users that are just viewing neurons in FlyWire. And on the bottom, I'm showing you the number of edits that we're currently seeing per day. The 7-day average, we are currently going over an edit a minute, which is quite a lot considering that these are actually manual inputs to the system. FlyWire has seen over 900,000 edits, and in this plot, I'm showing you a point per edit. And you can appreciate that most of these edits have been in the central portion of this brain, and that is what we have also been focusing on so far. About 37,000 of neurons that are in the central portion have been assessed as proofread, meaning they are mostly complete. There might be slight extensions, but they can be used for analysis. Over 50 labs have contributed, and there are quite a wide array of users that have contributed quite a sizable amount of edits. Over 120 users have contributed more than 100 edits. And this led us to believe that we are now ready to scale up to the reconstruction of this whole brain, or the proofreading of the whole brain. And because proofreading per neuron takes about 10 to 20 minutes, and so we can also now go and finish, basically fill in the cracks of what the community has not been doing so far, and proofread the remaining neurons, and then finish up the brain. Our first milestone is finishing the central portion, the central brain, which is doing the most computations in the fly brain. The neighboring sections that are not in the central brain that you can see here on the left and right, they're called the optic lobes, so they're mostly involved in visual processing. So the first step being finishing the central part. We believe that we will get to that point in the next 2 months and then release it later this year. And of course, the second milestone ultimately is we want to proofread all the neurons in this brain and make it available for anybody to use. FlyWire's technology that we have developed for that is universal, and our software tools are all available on the Sang Labs GitHub repository and free to use for anybody. And we are also very happy to help if somebody wants to set up their own system. And our technology stack is also already used for other data sets. There is another Drosophila community, FANC, which is basically a nerve cord. They have a nerve cord of a Drosophila. So we have a brain, they have a nerve cord. Essentially together we have a Frankenfly. And a larger data set is the microns data set that I've shown you one picture from earlier of a mouse spatial cortex that is also using the same stack, showing that we can scale to these very large data sets. And to end this talk, I wanna look ahead, what comes ahead of the fly brain. This was a plot that I showed you earlier. The fly brain sitting comfortably in an area that we can now handle computationally. The whole mouse brain is something that the field is striving for, and I believe we can reach in the next decade. It will take about an exabyte of storage alone, and we definitely have to improve our processing. And then quite out there, and I wouldn't make an estimate for when we get there, is a human brain that's 1,000 times more than a mouse brain. The mouse brain itself is 1,000 times more than the largest data sets that I've shown you earlier. And one of the challenges we have to overcome is something I want to illustrate you here. On the left, I'm showing you a fly neuron. This neuron is complete. We know we have all pieces of it, and to scale next to it is a neuron from the microns dataset. This neuron is not complete because the dataset truncates the neuron. It's only part of the whole brain of the mouse. It's about 0.1%. And everywhere where you see an end of the axon of this neuron, there is basically that's ending there because it is running into the boundaries of that data set. Nonetheless, that is one of the largest reconstructions from an EM data set ever created. And when we look at the proofreading times, we see that this neuron took about 10 to 20 minutes. However, this here took a proofreader 50 hours. So we definitely have a scaling problem right now to proofreading these large reconstructions. And if you want to scale further, we have to improve our segmentation, because I think what essentially happened is that we scaled the EM data acquisition faster than we improved the accuracy of our segmentation. And one way to look at that is that the CNNs that we are currently using to make these segmentations, they have quite a limited field of view. A CNN may see a few microns to make a decision to label in a neuron at a given location, but many errors are only apparent on a large scale. So here I'm showing you a segment that came out of the automated segmentation for the microns data set, and as a human, it's very evident that there's definitely one error in there because there are two cell bodies in the segment, but that is not visible on a small scale. So the next generation of segmentation algorithms will need to cope. We need to use larger contexts to find out, to see these biological constraints and understand errors better on that scale. Good. With that, I want to acknowledge the great teams that have contributed to all these data sets that I have shown. Chronotomics is a true team effort, and that makes it all the better. I also want to acknowledge the funding, especially from the NIH for the FlyWire, And with that, thank you to you for your attention. FlyWire has an open sandbox where you can try proofreading yourself. And I want to also point you to other public connectomics datasets. I mentioned the microns dataset before. This dataset has been made public last year, so it's fully available for you to see and explore. And the same goes for the human dataset. That's also a cubic millimeter. That was a collaboration between Harvard and Google, and also that one is available and ready for you to browse. If you basically just Google for HF1, you will see that one too. Right. Thank you, and happy to answer some questions. I saw that some came up through the talk.
Thank you very much, Sven. We have time for questions. I have loads of questions. I think it would take us, but one question I will ask: how do you associate physiology and topology of brain with function, and also biochemical activity, neurotransmitters and the like?
Yes, that's an excellent question. So in the fly, that is actually something that is more easily possible than it is in cortical data sets. And the reason for that is that the fly brain is quite stereotypical, so that means that if you have a connectome based on one brain, you can register that to another brain where you have done physiological experiments like light microscopy to measure activity of individual neurons. And this way you can associate activity or co-activity of multiple neurons with the connectivity that you extracted from a different data set. So that is something that makes the fly connectome even more valuable. And on a neurotransmitter side, of course, we want to know are neurons, are connections excitatory, inhibitory, and I mean, that's just the highest level of neurotransmitters. And different methods have now shown that we can actually predict these neurotransmitters from the EM images directly. There have been efforts, one effort in the FlyBrain, and this data is also available to the community at this point, where a group led by Jan Funke at Genelia, they showed that they can predict 6 neurotransmitters from an EM image alone. They had another data set where these were co-labeled, and yeah, so that data is also available for scientists to analyze. On cortical data sets, these things get a little bit more tricky because brains are not so stereotypical. So there you actually have to do the functional experiment in the same brain that you also then reconstruct the neurons in. And that makes these experiments extremely complicated. But this microns data set that I have referenced before, that has this done. So this is also one of the reasons why this is such a huge collaboration. There at the Baylor College of Medicine, they imaged a brain region while the mouse was running on the treadmill. Then this was sent to the Allen Institute. They reconstructed the EM stack, and at the Sang Lab, we reconstructed those neurons. Now we can associate those neurons with the activity, but that is truly a huge effort. And yeah, but it is possible to do that, and data sets exist that have that.
We have a question here from Krzysztof. How did you cope with anisotropic voxelization?
Yeah, so anisotropic voxels are make it more difficult, but in the end, these essentially the CNNs that we use mirror this anisotropy. So usually they have a bigger field of view, so to speak, in the lateral dimensions, in the lower one, in the lower resolution one, so that in biological space they about see the same. Anisotropy makes the whole problem more difficult. It makes definitely synapse detection for those synapses that are basically with like in a slice more tricky. But over the last years, people have built methods that can deal with that and train machine learning models to better handle these anisotropies.
Right. So we have a question: how do you cope with glial cells?
Glial cells are very tricky. They tend to be very intricate and very branched out. Our system on a backend can handle them because we also just partition them up into smaller pieces. But as it stands right now, at least in FlyWire, we tend not to proofread them because it's extremely tricky to proofread them, and we're focusing on neurons alone. And in the later step, we may come back to the glial cells, but yes, they are very tricky, definitely from a proofreading standpoint.
Sven, I will share some crazy idea with you. You see, okay. I often think how useless activity a lot of people sort of let's be really blunt. Lots of people engage in computer gaming, and in China alone, there's estimate there are about 150 million people playing games at any time. Mm-hmm. So I was thinking of embedding scientific tasks that require human intervention into the games as a task in the game. What do you think of this crazy idea? So for example, will it help your brain annotation?
I don't think it's that crazy because people have done that exactly that. So the idea of using games to reconstruct neurons, that goes back multiple years, and some of the at least thought predecessor of FlyWire is iWire, which is an online game to reconstruct neurons in a retina data set. So yes. It is essentially a gamification of reconstruction and proofreading decisions, though it is its own game. It's not embedded into another game. I have seen prototypes of games that are more removed from actually looking at brain sizes, but I have not seen them materialize and contribute in the end. Though that being said, I think there's a ground for introducing people in some sort of game to these data sets and use all that power that comes with it to speed up proofreading. And in the Sang Lab, we also working on opening up proofreading to gamers such as an iWire, and there are extensions of FlyWire that will do exactly that.
Okay, actually, we have a comment here from Bartosz that he commented on this EVE Online. I know nothing about games, but I think that would be possible. That's great. Now, the question from Krzysztof again. Did you contemplate some multimodal approach? For example, using high-resolution CAT scan, CAT tomography, or tensor MRI to gain some additional information?
So that is definitely a place I think the field is moving into, though those imagings are mostly done to help the alignment, actually. So doing a full CT scan, then to use that as an outline to align these cut slices in. So it's mostly thought to be used to help the assembly itself. I have not seen people put a mouse into an MRI detector. I think our data sets have been small on a scale compared to what these methods can resolve. I think our data sets are quite on the scale of a few pixels of the resolutions of these methods. But potentially, I think in the long run, if we can image very large parts of a mouse brain, it might be worth it to first put the mouse into an MRI and then compare what we get out of these two methods and align them.
Right. Sven, I have a question. Jan has a question, so I'll wait with that. Would it be valuable to reconstruct the connectome of the fly brain at different stages of development, perhaps to study dependency of neuron connections from gene expression pattern during the development?
100%. 100%. Yes. And I think the future of connectomics is going to larger brains. Another one is doing multiple brains of the same species and comparing them through development, through different disease stages, and to try to understand what changes in the wiring for these different modalities that we have. There are, I know, 2 works that have done connectomics through development, and I think that is definitely one huge avenue forward for the field. And I think a lot of insights will come from that.
So I have a question, Sven. Are there similar efforts for other organisms or insects?
Yes.
Let's say any other organism.
Yeah. So I mentioned the fancy community earlier. That is also a Drosophila. So the ventral nerve cord, meaning basically what is the spinal cord for us, is the ventral nerve cord for the fly. There is our microns data set is out there, and if people are interested in proofing that, we are also happy to share that and hope to also open proofreading for that going forward. I think so. There are other efforts that are not open to the public, but people definitely are reconstructing their data sets and starting to proofread them. And yeah, so I'm aware of works in zebrafinch, for instance, the songbird. It's a very interesting model organism, and some other work in C. elegans, and then a lot of work in mouse.
Right. And then finally, this question: when you have shown this wiring scheme, that reminds me very much, of mathematically speaking, to adjacency metrics. Is it if you look at different organisms and look at this from the point of view of graph theory, would there be some sort of mathematical features or that are similar, like I don't know, diameters, cliques, and because the Henry Markram work was on cliques, very interesting work on cliques. So could you distinguish some features of that kind here?
I think we could. I am not aware of this work having been done, but I agree that this would be extremely interesting on a graph level to see what are similarities between graphs from different brains and compare them. And I think one of the work that is now facilitated through releasing all this data to the public, is that people can go and do these analyses and run graph algorithms on this kind of data. And I think we are in the early stages of what is possible there. And the FlyBrain will also give us a start in building these methods, because the problems will only get larger from there. So yeah, I think that I think these questions should be asked, and I'm curious what we will find.
Thank you. Are there any questions? I don't see any questions, so maybe with this I will thank Sven. Thank you very much for joining us here and speaking from Seattle. Is it? You're in Seattle now.
That's right.
So it's thanks to internet and all this technology, we can be at the same sort of virtual space at the same time. So thank you so much. It's fascinating work. It looks like there will be more questions than the answers currently available, but that's the whole fun of doing your research. So I wish you all the best in your finishing your PhD and in your scientific career, and to your colleagues and your professor. Thank you very much, Sven. All the best.
Thank you very much for having me. And yeah.
Thank you. And let me finally remind you that this series of virtual ICM seminars is run with support of the University of Warsaw, National Information Processing Institute, and few information outlets: HPC-Wire, Datanami, Enterprise AI, ITWIS, and Forum Akademickie. And that was the 21st of our seminars. Here's the statistics. Here's the link to all the previous seminars, and I believe today's seminars will be available at this website within a few days, next week probably. And with this, I thank you all for attendance, and thank you, and have a lovely evening or day or morning or wherever you are. So all the best to you. Thank you very much. Bye-bye.